Hi, this is Kate and Lorenzo from MHC presenting about Bradi1g75350.3. Ab initio and homology-based models were both made for the gene. F-Ganesh predicted that it would have 8 exons and that it was on the positive strand, while Phytosome showed that it was on the negative strand and contained 11 exons. The log scale RNA-seq data also provided insight into the exon and intron composition of the gene. The highest log scale values were all associated with the exon regions of the gene, but the introns were also represented in the log scale RNA-seq data at lower levels, perhaps indicating that there are several different splice variants of this gene, or maybe this data was collected from mRNAs before they were spliced. Looking at the phylogeny, many of the related genes encode proteins involved in zinc binding like zinc fingers and zinc ribbons. When it was blasted, the unknown gene had 100% similarity to E3 ubiquitin ligase RZFP34. Based on the name, it may be an enzyme that catalyzes the post-translational modification of proteins by marking them for degradation with ubiquitin. The phylogeny illustrates that B. distachion's own RZFP34 gene was closely related to RZFP34 proteins in the other 4 organisms, with the closest relationship being with RZFP34 in O. sativa. The protein domains of the gene were determined through a blast that showed that the protein is made up of 4 different domains that involve zinc. Their characterizations are listed right here. Literature exists for RZFP34 in closely related species like A. thaliana and O. sativa, and these genes were confirmed to encode for the same zinc binding proteins. In one study done by Ding et al. In 2015, when the loss of function and gain of function muons for AT-RZFP34 were exposed to drought conditions and then rewatered, the loss of function muons continued to be wilted and didn't regrow, while the gain of function muons were able to regrow. The overexpression of the gene allowed the plants to recover, indicating AT-RZFP34 is integral to a plant's response and recovery from drought, allowing us to hypothesize that the function of the gene of interest is critical in the abiotic stress response pathway. When identifying the possible mutations in the plant, 5 were focused on: 1 high impact mutation shown by the red diamonds, and 4 moderate impact missense mutations shown by the yellow diamonds. The high impact mutation NAN0020 is a nonsense mutation that causes an early stop codon and is located in the very first exon. So a plant with this mutation produces a very small piece of the protein that the gene encodes for. The other 4 mutations change the amino acid at that location and have less of an impact on the transcript. The conformity of our primers to the target values for length, annealing temperature, and distance from mutation indicate that the primers should work well. When the DNA samples were quantified with the nanodrop, they had some irregular absorption values. A lower absorption value can indicate contamination that can impact the experiment. When the DNA samples were run through gel electrophoresis, you can see how both sets of bands pass the ladder, indicating that the bands are smaller than the ladder's smallest segment, suggesting a large amount of RNA molecules are present. In the second gel with RNase added, the 2 sets of bands that are present in the gel with no RNase were replaced by one set of bands that were very dim, confirming that the bands were RNA molecules in the first gel. After the PCR was run, another gel was done where there's a clear set of bands for each of the 6 samples, around 500 base pairs, indicating that the reaction amplified a region that was around 500 base pairs. The expected product length was 535 base pairs, so it seems that the PCR was successful. Nanodrop data was collected from the purified DNA samples, and the majority of the samples had very low absorption ratios, indicating large amounts of contamination.
So using these purified DNA samples, our next step was to determine where the location of our mutant site was within our mutant strains. So to do this, we first did a Sanger sequencing of all of our mutants. So we had 4 mutants, but only 1 mutant was returned that had a chromatogram that was relatively free of noise, and that is mutant 1. And we can see here there's a high or a green band, and there's only 1 big band. And we also conducted a pairwise blast for all of our sequences. And again, only mutant one returned a legible result. And in both figures, we can see that there's the presence of an adenine, and this is in line with our previous results in our previous data, which shows that the original wild type contained a guanine that was converted into an adenine, and this introduced a stop codon that likely resulted in the premature termination of transcription. And so our next steps of our investigation into this unknown gene was focused on determining the effects of this mutation. And so before we could do that, we first had to determine where the levels of expression differed within the different sections of our beta stachyon sample. And so we can see that the highest level of expression present using this map are present within the last internode in the first 2 centimeters of the peduncle. And so we can conclude that in general, the stems contain the highest level of expression. And our next step using that information was to conduct the phenotypical analysis of our samples, comparing our wild type stock to our mutant stock to see if there was any phenotypical differences between the two. We didn't expect there to be any because none of our previous literature and none of our previous data showed that the phenotype would be affected by our mutation. And our results back this up. We can see that our comparing this image of the wild type 2 versus the mutant 2, they're very similar, and any differences in brightness or coloration can be attributed to human error and differing methods or differing microscope conditions. And so to test our hypothesis um, that our gene is involved in the heat stress response of beta stachyon, which we made based on our literature search, we took a group of 8 young plants and we incubated 4 of those plants into, uh, we put them in a growth chamber that was set at 42 degrees Celsius, which previous literature showed is considered hot for beta stachyon. And we kept them there for 3 hours while we stored the other 4 at room temperature for 3 hours. And then we collected the stems from both sets of plants and we measured the RNA concentrations and compared them. And so using these samples, we conducted a gel to determine the size of the samples, or the DNA concentrations of our samples. And so using this gel here, we can see that we have 2 clear bands for all of our our room temp samples. And unfortunately for our treated samples, we have two streaks, and one where half of it is a band and half of it is a streak. Half of it is a streak, and so these streaks indicate that our samples were contaminated with RNase, and this is unfortunate. And we can see that room temp 3 or sample 3 from the room temperature group showed the brightest uh, samples. So next slide. And our next step was to use our treated samples and our control samples and to conduct a qPCR analysis to determine if there was any differences between in the expression of the gene within our samples. And so this first box and whisker plot, we we compared the relative expression in our wild type plants and our mutant plants, and these were both untreated. And we can see that the wild type plants showed a slightly higher level of expression, and the difference between the level of expression in the wild type and the mutant plants can be considered statistically significant based on this p-value down here, which was resulted from a t-test. And our second figure compares our relative expression in our treated plants, so the plants that were grown for 3 hours under heat conditions, compared to our control plants. And we can see that the treated plants showed a substantially higher level of expression compared to our control. However, we only had one out of three samples that returned a value from our analysis. So we cannot conduct a t-test, which means that we cannot conclude whether or not this difference is statistically significant. So although it appears that there's a difference in the level of expression, we have nothing to say that definitively. So next slide. And so our conclusions for our experiments and for our research project this semester was that firstly, expression of our gene is higher in the stems of our plants. We first hypothesized this based on our previous literature, and this was confirmed using data analysis of previously collected data from the Hazen lab. And our second hypothesis was that heat would lead to an increase in expression of our stems. And although we have data that appears to support this conclusion, we cannot say definitively whether or not it is true based on the lack of replicates of our treated samples. Again, we only had one treated sample that returned a result that we could use. And so for future experiments, we must repeat this experiment following the same conditions so that we can make any definitive conclusions. And next slide. And we would just like to acknowledge and thank Dr. Hazen, Nishadi, Kate, and Umas Amers for their help and support as we conducted these experiments and this project.